Friends, welcome to Agriculture channel. I am Varsha Gaitonde and here I would like to discuss about genome editing. Genome editing typically involves finding the part of the plant genome that could be changed to render it less vulnerable to disease or resistant to certain herbicide or increase yield, quality or other desirable characters. Genome editing is more successful in animal breeding, maybe in the field of medicine rather than in plant breeding. But recently many approaches are coming up in plants also. Researchers use molecular scissors to break apart the genome and repair it, which is a process that occurs naturally when plants are facing stress and can throw up new mutations that enable the plant to withstand further attacks. This evolutionary process can effectively be speeded up. It is possible to examine plant genomes in detail in laboratories and create mechanisms through which the relevant genes can be altered very precisely without the need of important DNA from other organisms. Use of genome editing first need to make transgenic plants with two components, that is an enzyme and RNA or guide RNA, and then combine to make desired targeted changes. The beauty of this system is after the desired changes have been made and identified, researchers can easily separate the transgenic plant or part from the edited targets by performing traditional cross or self-pollination. Coming to the historical prospects of genome editing. As you know, in 1977, the first integration of Agrobacterium tDNA in plants was done. In the similar year, TI plasmid identified for DNA transfer to plants, means it started with 1977 tDNA experiments in plants. In 1983, protoplast transformation was carried out in 1980, where tDNA found to be nuclear localized. In 1983, first genetically engineered dicots with foreign DNA found. The technique used here was carrot transgenic carrot plant was developed by that particular technology during 1983. In 1990, first fertile transformation of maize via bombardment or particle bombardment took place. Here, binary vector concept for plant transformation was also used. In 1993, first commercial GM crop was tomato. Then first fertile transformation of rice by Agrobacterium was done in the same year, along with transgenic herbicide resistant soybean was approved in US. In 1995, Bt insect resistant maize approved in US. Similarly, in 1998, herbicide resistant soybean commercialized worldwide, and with this, in the same time, widespread commercialization of genetically engineered maize, canola, soybean, sugar beet, carrot has taken. Then in 2000, Arabidopsis genome sequencing was done. In 2002, countrywide acceptance of GM cotton in India, even though cotton was popularized after 1994. Then in the same year, 2002, rice genome was sequenced. In 2009, ZFN and TALEN genome editing techniques in plants were introduced. In the same year, maize and sorghum genomes were sequenced. In 2012, ban on GM crop commercialization, that was ban of Bt brinjal. In 2014, CRISPR-Cas genome editing was introduced. Introducing of somatic embryogenesis during transformation technique was also found during 2014. In 2016, transformation of recalcitrant varieties. Then in the same year, resequencing of major crops and finding homology. This became popular because of the third generation sequencing methods. And furthermore, for achieving the food security, there is a scope for tissue culture free transformation, genomics assisted breeding refinements, then non-GMO genomics breeding, genomics based transgenics, then genotype independent transformation, along with improved varieties and hybrids and advanced plant breeding are the techniques which can together enhance the food production. Different techniques of genome editing are meganucleases, TALENs. TALENs means transcription activator effector like nucleases, ZFN, that is zinc finger nuclease, then CRISPR-Cas9, that is clustered regulatory interspersed short palindromic repeats. In the beginning, when genome editing technology started during 1970s, meganuclease and zinc finger nuclease were very popular. Even CRISPR-Cas was also identified during that period, but it became popular only after 2012 when it dragged the attention of many researchers. TALEN is a technology which was experienced and researched in Xanthomonas species, whereas CRISPR-Cas was worked on E. coli. It's a natural bacterial defense mechanism. When bacteria creates its own defense mechanism against the virus which infects it, that is bacteriophage. CRISPR is a site-specific mutagenesis technique targeting DNA which gaining popularity within 5 years and became a highly been utilized research tool across the world because of its precision and negligible side effects to environment as compared to genetically modified organisms. Multiplex editing, utilizing some techniques to target multiple traits within a plant genome, is also a popular effort among the scientists. Now India is at doorstep to accept this technology. The only requirement is conducting the crop trials and assess the benefits and side effects. There are several controversial issues like whether to consider the genome editing under the genetic modification technology or not, but many of the stalwarts and scientists are thinking that it is entirely different from the genetic engineering or genome genetic modification, because in genome editing it is not that a gene is introduced from some other organism, it is just modification of the certain base pairs so that editing will happen and we can correct the any defects inside the genome. In the research of cancer technology, AIDS and many other viral disease infections like Zika virus, Ebola and all those things, this is a very breaking revolution, we can say. Rather than giving much importance to political pressure or policy making issues, there is a need of overall decision of the country, including scientists, farmers, policy makers, non-governmental organizations, public-private research and development teams, and those who really think for the betterment of the country and accept the new technologies for attaining sustainability in food production. Genome editing technology paves a way in getting higher yields as well as for increasing the food production. Coming to differentiating the four techniques of genome editing, this video covers the differences between four genome editing techniques, then the procedure, as well as why genome editing could not get Nobel Prize even it is a breakthrough approach in the field of genetics. Let us see the differences first. First one is target sequence length. Meganucleases has around 14 to 40 base pairs. In zinc finger nuclease it is 18 to 24, in TALEN it is 24 to 59, and in CRISPR it is 20 to 22. Always remember, meganuclease and zinc finger nuclease, they always target the protein sequence, whereas TALEN and CRISPR-Cas, these are the two mechanisms which targets the DNA. CRISPR has ability to cut the single and double strand breaks or cuts in the DNA, whereas TALEN has cutting at a single base pair, means many of the talents are required in order to make the genetic manipulations. Components of these particular types. The specificity module in meganucleases is target recognition. In zinc finger nucleus, it is zinc finger domains specific. Fork nucleus is a gene or it's a domain. Then in TALEN it is tail domain, and in CRISPR-Cas it is cRNA or CRISPR RNA. The cleavage module in meganucleus is domain nucleus domain. Then in zinc finger nucleus it is domain. Then in TALEN it is non-specific fork1 nucleus domain, and in CRISPR it is non-Cas9 protein. Coming to the target recognition, the capacity of target recognition in meganucleases is low, whereas it is high in case of ZFN, TALEN and CRISPR-Cas. The cleavage specificity is high in all the 4 cases. Then off-target effect is found in meganucleases. It is also found in ZFN as well as TALEN, but off-target effect is less found because we are sending a guide RNA along with the Cas9 protein in case of CRISPR-Cas. The level of experiment in meganucleus, it is redesigning each target site it is difficult and needs expert in protein engineering. But in ZFM, the level of experimental setup needs expertise in protein engineering, and designing is difficult as in case of meganucleus. In case of TALEN, it is relatively easy, where designing of new target site. Then in CRISPR-Cas it is less accurate and very fast. It is easy, accurate and very fast. The method is to design the target site. Genome editing is like opening a book. Go to a certain chapter and a specific word and change the word or correct the spelling. It is nothing but the process of genome editing. Scientists can get rid of unwanted sequence of DNA from the genome, the total gene pool of the organism, just like a film or copy, edit or can erase erroneous or unwanted scenes or lines. Most popular of these new genome seizures are meganucleases. The design and construction is relatively cheap, straightforward and avoid the intellectual property barriers. These genome cutters are a combination of units to recognize specific regions of the DNA and an enzyme to cut both the strands of the DNA at a sequence determined by the genetic engineer. When the cell senses that this double-strand DNA break has occurred, it stimulates the cell's machinery to repair it. There are 2 possible outcomes. One is non-homologous end joining and one is homologous recombination. In any genome editing technology, these are the 2 basic mechanisms what we can observe. In non-homologous end joining, it means introduces a mutation at a site of cutting by genomic seizure following by natural repair process. Think that this is a double-stranded DNA and here a particular mutation is induced, and by genomic seizures this particular sequence is cut out, and by natural repair process means, as you know, the genetic errors are repaired by our DNA replication system only. Okay, there are several enzymes in order to do this, and by this repair process, that is nothing but non-homologous end joining. Okay, the second process is homologous recombination. Here a separate DNA molecule that has the same region in it as the region that is trying to be modified in the host genome. It also contains a gene coding for the desired additional trait. Here what will happen? Additional DNA is introduced. That means here just by mutation it is cut open, and by the repair process this is recovered. But in case of homologous recombination, the portion which is not required, it is cut out and separate DNA sequence is introduced. This by complementation the new sequence will be recovered, and that will recover the double strand, and particular defect is overcome in this way. By using these methods, gene can be knocked out, that is silenced, or new DNA including whole gene units be inserted. This is the process how it goes. First one is designing and synthesis of target sgRNA. This is an example in CRISPR technology where single guide RNA is designed. Okay, guide RNA is for the purpose it guides the Cas9 protein to get into and binds at a specific position so that the modifications can be done. Then genomic target is set up. Use online tool to minimize off targeting. That means already there are several databases where the genomic sequence is available. By utilizing that, we can get the genomic targets. By that, off-site effects can be minimized. Then assemble a Cas9 sgRNA construct. That means we can construct a plasmid vector so that it can be inserted into the plant system. Then go for cloning of the construct in suitable plant binary vector. By using it in a vector system, we are just cloning means making it into different or many copies or multiple copies. Then introduce this into a plant by agrobacterium mediated transformation or any other gene introduction techniques. It may be particle bombardment, gene gun, or anything. Delivering into plants, here the techniques are given, maybe agrobacterium, protoplast transformation, or callus bombardment. After that, the regenerate and screening transgenic plants for genome editing means that should be checked whether that particular gene is properly acting or it is properly functioning or not. After that, agrobacterium mediated transformation of plant viral delivery of this is checked. How? By selection of non-GM genetically edited plant with desired modification. That should be checked whether it is properly inserted or not, whether it is functioning properly or not. This is the entire process how the genome editing works inside the plant. It follows the same principle how the genome or genetic engineering goes, but in genetic engineering, as you know, we introduce a foreign gene, but here it is no, it's just a sequence which can go and bind and alter the particular sequence. If the technique is used for correcting the defects, means the technology is called as gene therapy. Coming to the application part of genome editing, it's highly efficient in making site-specific integration and gene stacking. As I told you and showed you in the figure, it is helpful in site-specific integration, means integration of a particular sequence at a specific site, and gene stacking. Gene stacking means genes responsible for different traits are stacked or brought together at one place. In the process of gene replacement via homologous recombination, I told you already. Here what they will do, I'll show you here. The DNA strand is there, and by cutting this particular site which is unwanted or it may be having certain defects, a new DNA sequence or segment is introduced here, and by homologous recombination this double strand can be recovered. Okay, then utilize the chromosomal engineering through which site directed deletion and translocations can be done. Labeling and modifications of multiple genomic sites is possible through editing Okay, when we talk about genetic engineering, we don't know whether it will be successful or failure, because when a new gene is introduced in a genomic system, it will disturb the entire. As you know, if we are injecting something into our body starts reacting. Just like that, when a gene is introduced into a plant cell will start counter attacking that particular gene. Likewise, whenever we talk about this modifications, it is quite difficult, but here we are just altering, or we are just removing that particular site and going for the repair mechanism. This won't cause much harms as the genetic modification do. In genetic engineering also, the defects what we observe, or we can say in common terms, the side effects can also be overcome by editing technology. Transcription modulation of multiple genes and pathways are done by this. In epigenomic editing, such as methylation and demethylation, can be controlled by editing technology. Epigenomics means where the changes are observed at phenotypic level but no changes at DNA level. Okay, this is applicable in cisgenesis without introducing target foreign gene. As you can think that cisgenesis is better than transgenesis. In transgenics, the gene from any organism can be introduced, but what is in cisgenesis is here only the relevant species or genera can be considered in order to transfer a specific gene. Likewise, many of the countries, the regulations classified genetic editing in the category of cisgenesis rather than transgenics. Reducing cost, which makes molecular breeding cost effective. It helps in eliminating or significantly reduce the regulatory requirements. It is the only accurate technique to replace the genetic engineering crops and satisfy the public concerns. Coming to the application part, in rice, by editing gene or target LAZY1, by this tiller angle was improved character. Likewise, OS SPD for changing plant color, OS sweet 14 for bacterial blight resistance. Then in case of maize, IPK1 for non-phytic acid, UBI MOPAT, that is for chlorsulfuron resistance. Then in maize and sorghum, 4Cl2 for reduced lignin and tannins. Then in potato, for vascular invertase enzyme was recovered by prolonged color tolerance. In coffee, there is xanthosine methyltransferase silencing mechanism that enhanced the coffee without caffeine. This is a major breakthrough in the field of science, because caffeine causes several difficulties in body, like it may cause severe headache or many problems like nutritional imbalance or mineral deficiency. In case of Arabidopsis and Nicotiana, by the edited gene was S gene knockout. By this, disease resistance was achieved. In soybean, FAD2 gene, the character improved was for oleic acid and reduced trans fat. In wheat, HLA DQ2 and DQ8 silencing, the character was reduced gluten content. Then in case of banana, vitamin A improvement and fungal disease resistance, which is ongoing research. Then in various plants, for mutated gene or creation of variation, several gene addition or deletion or gene stacking was taken up. Let us see why CRISPR technology did not receive Nobel Prize. There were several reasons. One important reason is several patent issues are involved in the development of technology and the sequences. As you know, the CRISPR chips are already available in the market. The sequences will be given to you, and by using it in your laboratory, you can alter any sequence which you want to do. Likewise, many people or many companies are preparing these chips, and there are many patent issues. The second issue is safety issue, and exact impact on the environment was not clear. there is a conflict between whether it is considered to be GMO or non-GMO. Then in 2015, three scientists, they were Lyndall Thomas, then Magic Paul and Sankar Aziz, awarded with Nobel for their contribution in mechanistic studies of DNA repair, which is one of the process in CRISPR technology. For that, the Nobel Prize committee was not ready to give again in 2016 for the same work like CRISPR technology, which was very popular since 2014. But CRISPR was awarded with several other very attractive prizes across the world. Thank you. I hope you understood the concept, and don't forget to share, subscribe, comment and like. Thanks once again.